Hello, this is Tom Kalura, and I'd like to welcome you to our symposium. With my co-authors, we will be describing our construction work, which is very much a work in progress, and validation of a platform that is shared between colleagues for event-related potential work. I'm very happy to be also welcoming Thomas Feiner and Christoph Garner and Harry Karasides to this symposium. I'll be giving some introduction and then allowing them to share their very interesting work. First, I will disclose I'm the founder and president of Brainmaster Technologies, but none of the scientific content of this presentation is specific to any particular products or services. We show multiple ways to achieve the scientific goals that we talk about. Kind of as an overview, we're going to talk about event-related potentials being time-locked to an event or a stimulus, and we always require some sort of signal processing due to a low signal-noise ratio. This is a common shared aspect. We're going to look at different methods, including averaging, filtering, and some advanced methods, single response, for example. We're going to think about self-learning as a future possibility. We'll also look at steady state evoked potentials. And the application areas include assessment, but also neurofeedback, and also in counseling and therapy, ways to address the electrical data we're getting in a clinical context. The goals of a shared ERP platform are that we have flexible stimulus control, a variety of stimuli, standard file formats, some standard stimulus marking methods. These are just very important key aspect, and we want to ensure timing accuracy. We would like real-time response, also the need for post-processing for further analysis, cross-platform compatibility, and also user configurability for innovative methods. Our goal is not any particular type of ERP, but to make it possible to do any types of ERPs. So what is an ERP? It's an event-related potential or field measured at the scalp, and through repeated measurements presenting of a stimulus, we can find that the response, as though it's lost in the noise of the background EEG, nonetheless gives us the ability to see the repeated response and use averaging or other methods in order to tease out that response. One of the standard methods is to use a checkerboard, and there are clinical standards for using checkerboard reversal. And a lot of the work we're doing will use checkerboards, particularly for sensory evoked potentials, but also as a standard method that you can achieve on a computer, as well as with special kinds of equipment. So the visual evoked potential, for example, there are early components that are very interesting that are within a couple hundred milliseconds. And we'd like to be able to measure those quickly and accurately. They tend to be more sensory related and are very important diagnostically in terms of symmetry, etc. Now, if we have a more complex plan, such as rare and frequent tones, we now start getting cognitive evoked potentials, and this shows that the difference between the common and uncommon shows up in a later potential, and this would be the P3 or P300. These are also very important diagnostically. So when interpreting these ERP waveforms, we think in terms of the regions of the brain that are being activated. These can be then subjected to inverse solutions and more detailed mapping. But we do know that based on the physiology and the principles of how we present the data, we do get waveforms that represent constituent components generated in the cortex by cells of either a sensory or of a more of a processing, even motor-related potentials can be recorded. So the goal of a real-time evoked potential, which is one of the more difficult goals, is we would like to do assessment and visualize responses to specific stimuli, both sensory and cognitive, visualize brain processes over short intervals. We'd like to identify microstates, transitions between microstates, and possible candidates for feedback if we have sufficiently instantaneous ERPs. It may require the system tailored to the individual, such as learning that person's background EEG or learning their dipole configuration. And it may or may not be task-oriented. You can have ERPs where a person is just viewing something, and you can also have ERPs where there's a decision being made. And that can be a decision to press a button, or it could be a decision not to press a button, in which case you now get some very interesting inhibitory ERPs. So the standard methods are kind of contrasted here. When we do QEEG maps, we just take at least a minute, but we just record the EEG. We get overall amplitudes, connectivities over the entire epoch. It's generally not event related in any way. And the QEEG in particular disregards or distorts morphology, the shape of the waveforms. When we do averaged evoked potentials, we need a typical number. 32 would be a number, and that requires a certain amount of time to provide all those responses. And it mixes different responses into one standard form. And you could use a sliding window. We'll show you you can use filtering, and you still need accurate stimulus timing and recording. So the difference between QEEG and evoked potentials is really very profound, very basic, a lot of differences. So alternatives, if we want real-time evoked potentials, is we can do signal responses. They are obscured in background activity, but we could superimpose them, or we could use waterfalls or overlaps to look at them. And it would provide a time resolution of the sampling rate, which would be typically 4 milliseconds or even less. Now, another method which I'm going to show, which we have used for almost a decade, is event-related brain activation imaging. And we use the full scalp and S. Loretta in order to image the entire brain in all the frequency bands. This gives us 125 millisecond time resolution. We can coordinate with the stimulus presentation. It easily adapts to an instantaneous display, and we can get surface, S. Loretta, or Z-scores in real time. This brain activation imaging is amenable to real-time applications. What is done is that the responses are recorded on all 19 channels, and we do record event markers. And then based on the events, we simply look at the maps in 3 dimensions at all different frequency bands and look for activation patterns instantly to single responses. Now, this turns out to work pretty well. These are examples of maps which are generated 8 per second. So these are 125 millisecond spacing. And these are responses to different words that a person is listening to or reading on a screen. And as you can see, these distinctive patterns of response in the frontal hemispheres having to do with gamma activation, very different and actually very relatively repeatable within an individual. So these are all very useful in that context. Here's another example of a large number of these running together. And you can see the responses. It can be very complex and last for several seconds. And these are single responses. Again, there's no averaging needed to create these. So this is an important option for this type of work. Now, if we do averaged EEPs, this goes all the way back to the 70s, and we can show averaged evoked potentials. These are to repetitive stimuli at different frequencies, 2 hertz, 3 hertz, 4 hertz, etc. And you can see the repetitive response does give you a definable potential, a stereotypical evoked potential. And these would be done on something like a signal averager in those days. And this can be adapted also to a real-time measuring approach, real-time steady state visual evoked potentials. And this is a method that I happen to have used in my lab with others in the 70s to study responses to light. This was all done on an analog computer. So this is how it was done in the 70s. There's no digital computers here, just analog machines doing all of the transformations. And what was found is that the filtered responses were matching the averaged responses as well. So filtering becomes an alternative way to measure evoked potentials in real time as an alternative to averaging. Now, here are filtered responses to different types of vigilance. And these are 2-minute recordings. And under visual vigilance, we see a certain stereotypical response in the evoked potential components. These are the fundamental and harmonic of the stimulus, auditory and visual stimulus. And this is under visual vigilance. And this is under auditory vigilance. So now you see the response has a different time course, and the person is under a different vigilance state. So this again becomes a form of real-time feedback that can be used as for brain evoked potentials. The steady state evoked potential can also be applied for BCI, for brain computer interface. Here's an application by Scott Maycig showing the use of independent components analysis and frequency analysis to tell where you're looking on a screen. So again, this is kind of backwards. This is taking the signal and trying to control a computer. We're interested in using the computer to help the brain self-regulate. So conceptually, we'd like to take a signal averager like this, and we'd like to get it included in an EEG system of a modern digital form. And so with software, you can do that. And one of the things we've worked on the platform is an instant analyzer, which does the averages in real time and applies it to all the channels. You can have a 19-channel EEG now and do the averaging using the signal averaging capability. And here we see the, here, this is a video actually. And you do see it starting the engage there, and now it's beginning to extract the evoked potentials. And this is a real-time demonstration that while you're gathering the EEG, you can also be calculating these evoked potentials and seeing them generated in real time. Here's an N of 3 for uncommon and an N of 8 for common. And then clearly there's a difference then between these different response types. And it's possible then to review the instant evoked potentials using the file storage format so that then they can be reviewed later, as well as having the raw EDF files with markings are obviously also stored and accessible for future use. A typical control panel for ERPs then has all sorts of different options to pick the channels and the types of stimuli, types of feedback, all sorts of stimulus parameters. So those parameters, which are very specific and need to be defined, has been set up so they're basically optional. And the system that Tom Feiner is going to show also has a tremendous amount of flexibility in that regard. So when you're doing work on a computer platform, you must ensure that the stimulus is synchronized. And for example, with this checkerboard, we've shown the use of a photic detector, which is stuck on the screen. We left the cable there so you could see it right across. And it measures then the state of the screen and becomes yet another piece of data. So this is another reason, again, that event-related potentials tend to be much more complex than just doing QEEGs or EEGs. You can verify the synchronization, and this becomes very important. In this case, we're comparing an internal stimulus clock with the external measurement from the photic detector, and that we're seeing there's a consistent latency inside the computer, but then a very consistent response that we can then visualize. And this is part of the whole process that's necessary in order to ensure that we're synchronized and getting valid data. You then can verify. We have, for example, here's a P100. And so at 100 milliseconds, we see that pronounced peak. It's at 85.6 milliseconds, and it's 4.86 microvolts. So it's completely within the normal range. And along with that, we show the stimulus data so that we're sure that the stimulus actually has arrived at the time we think in order to make that very precise measurement. We can also use Fourier Harmonics. Here's an example of the filtering concept, which we're beginning to use a little bit. Here you see the stimulus pulses, and beneath it we have it represented in all frequencies at 2 Hertz intervals all the way up to 24 Hertz. And so you see there's some content at some of these harmonics, odd harmonics actually, and not so much on the even. And on the bottom you see the reconstruction. This is the waveform reconstructed from all these Fourier harmonics. So it's a very nice method to get that information. And here, for example, we're reconstructing some waves which were picked up from the sensors. And again, you can see that by using harmonics, the system is able to reconstruct these waves. And in the presence of noise, this would be an efficient method because those filters would tend to reject uncorrelated noise the same way that averaging would. So the options, these are just examples. I want to emphasize the options for presenting and processing, and we want to support all possible methods. So you can have checkerboards and images that are built into software. There's external software such as ePrime, presentation, SciTask is another one out there. Capito is produced by IFEN, and Tom Feiner will explain that. Excellent Brain will be explained by Harry Karasides, which is a cognitive and concussion assessment method. And you can use PowerPoint and other methods to present data. The processing can be built in using averaging or steady state filtering. It can be processed in MATLAB or EEG Lab or the IFEN ERP analyzer, Excel, Access, etc. I'll show some examples. With Dr. Karasides' task, he gives target and non-target images using different emotional faces. I'm sure he'll describe some of that. And when we look at the responses, we can see across individual responses certain systematic responses. This is done using MATLAB. So we've linked between the Excellent Brain application, then using Brain Avatar Discovery, and then using MATLAB EEG Lab, and we're able to get nice evoked potentials, single responses, as well as here are independent components, ICA, independent component analysis, using a special GUI, a graphical user interface, which is basically these buttons in order to control that package. So with this sort of wrapper or user front end, MATLAB EEG Lab can be used to analyze basically any ERP data that has the markers that we look for. Here's an example of a P300 difference in MATLAB. And here are some single responses lined up, and you can see that time goes from bottom to top in terms of the number of responses, and the time axis is left to right, the ERP itself. And you can see this is about 50 responses, and you show systematic changes from one response to the next, but they're very consistent. So this is important single response data. Here on FP2, again, these are for target faces, and you see based on the face, some of them are happy, then they're sad, then they're neutral, then they're angry, and you can see systematic changes right across single responses using that very powerful paradigm. And as we showed, the instant ERP can be used in a playback mode to get the evoked potentials. Here are the same potentials in the IFEN ERP viewer. So again, we have this cross-platform compatibility showing basically the same results and validating each other. So as a summary, I want to say we have a flexible platform. We're talking about multiple formats, activation imaging, steady state evoked potentials, single response evoked potentials, averaged evoked potentials. The components are interchangeable, and the different components are stimulus, acquisition, processing, and post-processing. They validate each other through cross application. This is configurable for any paradigm. If you want to do contingent negative variation or mismatch negativity or inhibition responses, it's all possible. Cognitive assessment for ERPs or for sensory and cognitive. You could take 1 to 24 channels, and that includes biofeedback capabilities as well. So I'm just going to show some references, some of the material we look at, talking about state machines, evoked potentials, microstates, etc. And with that, I would like to thank you for your attention and very much welcoming Thomas Feiner and Christoph Garner and Harry Karasides in their presentations for this symposium. Thank you very much.
Hello, my name is Thomas Feiner. I'm the founder and the director of the Institute for EEG Neurofeedback in Munich. Our institute has become one of the leading providers for neurofeedback education. And in our own research lab, we develop tools and programs for trainers and researchers in the wide field of applied neurosciences. Our most recent project was to create a system which allows to measure event-related potentials in a much easier and more comfortable way.
I'm now showing you the system we have developed in our lab, which can present auditory and visual stimuli with minimal effort. So even beginners in the field can have an easy start with stimulus presentation using the system for ERP measurement we have created. But also advanced users will benefit from our system. The jitter produced by Windows cannot be accepted and generates inaccurate results. Therefore, a technology called transistor logic is needed to get accurate data. Our system addresses cognitive event-related potential at first, like measures for P300, MMN, FRN, ERN, CNV, and others. The system consists out of several hardware elements, mainly the trigger box and the photosensor, which is here on the left side of this picture. And on the bottom, you see the trigger box with the push buttons for motor response or for testing purposes. And on the left, there is a photosensor with two photodetectors, which are attached on a monitor, which you see here on the right side is the photosensor attached to a monitor. And underneath, you see the signal and the markers that are produced by the presentation software. These markers indicate trigger information for the photosensor. The photosensor will send the recorded information to the trigger box, where it then is produced, where it produces electrical signal and sends it to the EEG device via cable. So the result is a minimum of jitter and a maximum of accuracy for obtaining accurate results for the triggers and for the averaging of the EEG, which is done later by our ERP analyzer that will be presented by Dr. Garner, who is one of the developers of the ERP analyzer in our lab. This is the GUI, the graphic user interface of our trigger presentation software, of our stimulus presentation software, which when presentation is started, it sends the trigger marker simultaneously to the screen and where it can be detected by the photosensor. These markers then are sent to the trigger box and then further to the EEG device. I have already shown this in the previous slide. Here we have some buttons where the user can just use predefined stimulus presentation protocols, but there's also many options to create the optimized individual stimulation protocols, which we will see in the next slide. So one of the very simple things you can do with this tool is just to create a simple visual oddball paradigm. And here is how it looks like in the screen of the software in the graphic user interface, where you have several options. And let's look at the top here. There is a picture presentation selected, but in the drop-down menu, you can also select visual, also auditory stimuli for presentation, like tones and sounds and so on. The colored boxes, we call them boxes, are places where where several stimuli can be chosen, and clicking on the plus button here opens the stimulus library for pictures in this case, and you just select the pictures you like to present in the protocol, and then it will appear in the box for common stimulus, or in the box for the uncommon stimulus. Here it is said we clicked on the green box, and then we select the stimulus for the common stimulus. If we click on the red box, we select the stimulus for the uncommon presentation for the rare stimulus. So, and if you click inside of the white box, you can then select and choose a picture or a stimulus from the library, which then will present a neutral stimulus. This is important because the software from this box will just know, the software will know which marker it will produce. The marker on the screen that where the photosensor can read the marker information, whether it's an oddball or a frequent stimulus. Okay, and further down we have the presentation, the number of trials of the presentation. We have chosen 130 in this case. Number of trials, that means 130 times there will be a stimulus presented, and it can be a common or an uncommon stimulus. And here we define how often the uncommon stimulus or the target stimulus should appear. And here we decided it should be 20% of the whole number of the presentations, and it will be there will be 26 presentations of the uncommon stimulus. Also, the trial duration, of course, can be defined, and here we have chosen 1,000 milliseconds. It can be, of course, more or less, and there is a stimulus duration you can define, and well, there can even be an interval between the trials that can be chosen randomly. Here is a possibility to choose random, and then all these stimulus presentations will come randomly. Okay, we call this also a jitter in between the trials. This is a very simple way how to set up an oddball stimulus protocol with visual or auditory stimuli. The next thing we look at is the single stimulus creation of a presentation, and this is done in this way, where we still have the same boxes where you define which stimulus should have which characteristics. Here we choose the O letter as a common stimulus, and in the next, when we saved this stimulus presentation protocol for this one stimulus, we just switch them to the uncommon stimulus, which is then shown here as the Y button. Shoot. And the same way it can be done with the uncommon stimulus. We click on the box where we want to place the uncommon stimulus, and we select the letter Y picture from the picture library, and we can use the same setting, 1000 milliseconds for trial duration, 500 milliseconds for stimulus duration. There is 100% target, of course, because we always want to see the target when we only want to have one stimulus, and there is no interval in between. But of course, in the countdown, you can define a certain onset time so that there is a pause between one presentation and the other presentation, or the one between one trial and the other trial. So now we have two trials defined, one for the common, the other for the uncommon, and we can also create a stimulus protocol using the single trials we just defined. And here on the left, you see that we already have a collection of single trials or of tests, and we can use them always later in our session protocols. And the longer you work with the software, the more possibilities you have collected, and then you can create protocols from a collection of trials you have already in that folder. Okay, then let's go to the next slide. Then there is the presentation of sessions, how you can create sessions, which has more complex presentation structures. Okay, and here you see left there is the structure that consists out of 11 different trials, and one trial is a sample shown. Oh, sorry, there is a sample shown. There is fixation cross. There is a match picture. Then there's another fixation cross at a different duration with 2,250 milliseconds. Then there is another match picture, and this is actually one of the tasks we are working with, at the moment on, so-called delayed match to sample task. And in the session list, there can be like 100 or 200 or more several trials and single trials that are defined in the test section, which some also call the stimulus library or the stimulus list here. But this is not just a stimulus, it's those stimulus presentations are already defined, whether they are a common or an uncommon stimulus or a neutral stimulus. Okay, and the session task, the test sections inside the session can also be very well be modified and edited. You can add other trials or remove trials or change the order of the trials whenever you need to do this, or whenever you think you need to optimize a protocol, or you found an error in it where you want to remove or replace several trial which you found which is not working or which doesn't fit your purpose. And of course, you can assign presentations to subjects, to several subjects, which is also a possibility inside the software, where you can define a subject name or create a subject folder in which you can add other stimulus presentation protocols. The meaning of event-based potentials for clinical research and practice increased, and research shows the value of such findings. ERPs present promising tools for evaluating the biological source of functioning of the brain in many neurological and psychiatric disorders. And here are some examples of research and findings which we think are very important. We think because of this, it's about time that the enormous potential of this method will be more and more established in clinical practice with easy-to-use methods for both the researcher and the clinician. At the end of my talk, I want to thank all the great people in our team and the collaborators who helped us so much to move forward with this project. And thank you very much for listening and your attention.
Okay. My dear audience, my name is Dr. Christoph Gardner. I am a neurologist, and in the last years I have developed together with Mr. Feiner and the Institute IFEN a program where we can analyze event-related potentials, ERPs, with on a much more simple manner than it was possible before. The program was initially written in MATLAB, but then we transferred it to Python because there were more options we could use there. Especially we could compile it and give it to others simply. So to show how it principally works, we only have to do a few clicks. We have to load an EDF file, EEG recording with 4 channels, 19 channels, whatever, and open this. And now you can see we have a 19 channel EEG, and we have EEGs triggered on the uncommon event. In this case, it's a visual oddball task with O as an uncommon rare trigger and X as the common one here. Yeah, to see that the ERP there, you only have to click on the channel you want to see. Maybe commonly first we are looking at CZ at the central on top and say run analysis. And then you have the averages of the epochs of the uncommon. This is the red line. Yeah, there were 6 triggers, and for the common event, the X, these were 38 triggers. And we have a clear P300. This is a healthy person here, young person with about 30 years. And the P300 we can see here is it's 223 milliseconds. Here you have the scale for the amplitude. Here you have the scale for the time. And we have a latency of 324 and an amplitude calculated from 0 of 16 microvolts. This is for an ERP, it's quite large. So how do we come to this picture? I want to show you first the original CZ EEG together with the triggers. You have a button down here, show selected EEG channels, and now we are showing the CZ EEG plus the triggers. Yeah. Now you can see the red lines are the triggers, and the blue line is the EEG. It was a recording of 2 minutes, 120 seconds, and as I told you, about 40 common and 6 uncommon triggers. We can enlarge this to show you the principle of Mr. Feiner's work. We have here triggers, and now I am moving through this. These are all small triggers with about 60 milliseconds. And now we go here, and now we see here is a broad trigger. That means an uncommon event on the screen, an O. X, X on the screen, X, X, and here's again an O. Yeah. What we are doing now is that we average, we cut 1 second from the beginning of the trigger, yeah, and put it into a basket. If it's a broad trigger, it comes into the basket uncommon. If it's a small trigger, it comes into the basket common. And at the end, we divide through the number of triggers, and then we have the average. We can do this now, and we can see this is the uncommon trigger, 6, yeah, and this is the corresponding averaged EEG of 1 second. I can make it more clear if I go for plot normalize, and then you can see the P300 at 330 milliseconds here. We can do the same with the uncommon. CZ is here, and we again also do the uncommon, do the common trigger, and here we have the common trigger, and if I do normalize, we can see at 300 milliseconds there is no potential, as we could see here if we do both channels together. So what can we do now? We can, for instance, we can subtract the 2 channels and see what is the difference. We have here options and tools. We can make, we can clear the channels. Yeah, for instance, if I want, I can clear markers, and I can clear the screen if I want, and then I have an option difference. And so this is the difference of uncommon and common. Yeah, we can also do show all 3 lines. So this is the common, the blue one, the red one is the uncommon, and the green one is the difference of the 2 lines. So next, what we like to do is to make this the curve more clean. So you see there is a lot of spikes and high frequency amplitudes. And what we commonly are doing, we are using a low pass filter. That means now you can see here 20, we will filtering out all frequencies above 20 hertz, because our potential is a slow potential. Yeah, if I do this, I get a much more clear line where I can more easily identify the amplitudes. Next, what we can do, we can also use a high pass filter. That means we are cutting out all frequencies below 0.5, so slower than 0.5 hertz. This is especially helpful when we have a lot of amplitude changes. I will better show you this in the EEG itself. If I have no filter, sorry, if I have no filter, you can see this is 0 here, that the ground line is shifting up and down. And if I use this high pass filter, then you will see the EEG will be much more flattened. Yeah, you can see all this up and down, the sweating effect, something like that is killed out. So regularly we are using, optionally of course, high pass and low pass filters, and have a nice curve with the N100, a P100, P200, P300. This might be a P4 or 500, things like that. Sorry, I moved this. You can enlarge any interesting portion if you want. You can have a close look at the peak. You can also wander through the curves with this button, and there are some other options also. So what I want to show you next is what we can do is we can, of course, now look at different channels simultaneously. For instance, how is the propagation from back to front? Now we have in blue the C set, blue one, in red P set, posterior, and in green we have F set. Yeah, and you can see all three are quite the same. As I told you, you could even look closer to it if you want, for instance, like this. Yeah. Something else we can do, we see that these curves are starting all little below 0. We can make an adjustment, start with 0, so every curve is here normalized to 0, and some people like to calculate the amplitude in this way. Another way would be to do a baseline correction. That means we are averaging 200 milliseconds before the event and adjust the lines to this average. So this gives a little different picture there. What we can do, of course, is now also to look, make clear channels. What is FP1 and FP2? So right and left side. What is it looking like? Very nice P300. if we look at the common, I will predict that the common will run here, somewhere here, yeah, because there should be no P300. I will add it, yeah, and you can see the common event, the X event. There is no P300. Last, I will show you if we analyze or we show all common channels together. You can see these are all channels from front to back, from left to right. And what we can do now is to find the amplitudes here and make them graphically. That means I'm looking for the amplitudes between 250 and maybe this one would be 380. I can put this number here, 380. Yeah, and then I can export to an Excel table. Now I have to change the screen, I think. Yeah. So now you can see a map, a brain map of the latencies and of the amplitudes. The green values are faster P300s, and the yellow and red ones are slower ones. As you can see, this individual has faster latencies on the right side in the average of about 10 milliseconds. Yeah. The amplitudes, however, are normally distributed lower latencies back, higher latencies in front, but the difference, side difference is 1 microvolt, so nothing. Here there are more detailed figures about the differences of the latencies, and in the bottom here you have the all latencies of the P300. They are mean 331, and the amplitude is a mean 9 microvolt, what is quite good, and the standard deviation. Yeah. So that is a short introduction into our ERP program, and I hope I could show you that with this program, it is possible with a relatively simply and fast way to analyze ERPs. Thank you very much.
Hi, my name is Harry Karasides, and I'm presenting today on the simultaneous cognitive performance and event-related potential measures in sport concussion management, an integrated approach model. As a matter of disclosures, I'm the major stockholder of Concussion Management LLC. I'm the author of Concussionology, redefining concussion management for all levels, and I'm the medical director for Stress Therapy Solutions. Concussion is a brain injury. It's defined as a complex pathophysiologic process affecting the brain induced by biomechanical forces. This is one of many definitions. This one was provided by the Consensus Statement on Concussion in Sport in Berlin in 2016. By and large, all of the definitions really define concussion as a functional problem with the brain rather than a structural problem with the brain. And notice that features such as loss of consciousness are not part of the definition of a concussion injury. It's a very common condition. Approximately 2.8 million traumatic brain injury cases present to American emergency rooms, hospitalized or even die from traumatic brain injury each year. Over 300,000 pediatric sport and recreational ER visits occur due to traumatic brain injury. And recently, the CDC estimated 2.5 million sport-related concussions in high school athletes alone in the US. In those athletes that play 3 or more teams throughout the year, there's a 1 in 3 risk of having a concussion. In the US, the highest incidence of concussion injury is in football, American football. Second to football is women's soccer. And in girls soccer, actually in any sport in which girls and boys or women and men play by the same rules, women tend to have more concussion injuries than men. Third in line as far as concussion-prone sport in the US high school arena is boys lacrosse. To give you some background, currently, the evaluation of brain physiology is not indicated or not included in guideline recommendations for concussion management. Numerous national and international groups have provided concussion guidelines for concussion in sport, for management of concussion in sport, and none of them include measurement of brain physiology as part of the guidelines. We propose that QEEG, and specifically S-Loretta analysis and cognitive evoked potentials, are well suited as an independent, objective measure of brain function to provide insight into the pathophysiologic process of concussion injury and recovery, to assess injury severity, and to track recovery. This little video, I love this video because it really highlights the forces that are applied to the brain through biomechanical force, and highlights the injuries that can occur due to the direct trauma, which is typically the coup injury at the point of impact, and the contra-coup injury, which is the recoil of the brain as it moves backwards away from the point of contact. Shearing forces, which are typically due to these pressure waves that stretch and compress the brain in its gelatinous form. And a diffuse energy crisis, which occurs because of the high demand of energy for repair and also electrolyte balance, and the low supply of glucose, which is affected by the dysfunction of the glucose transporter protein that is embedded in the neuronal cell membranes. There are a few unique features of sport-related concussion. There's an implicit or explicit acceptance of risk, which is typically accepted by parents rather than by the youth athletes. And this highlights the importance of awareness of this particular risk. There's also a phenomenon of risk compensation in which athletes that are suited up with protective pads, protective gear, helmets, even mouth guards, can lead to a psychological phenomenon in which the athlete will take greater risk than if they were not wearing this protective gear. And that leads to a higher risk of having injury. This phenomenon has been demonstrated even in cyclists who wear helmets versus cyclists who do not wear helmets. And in some cases, the athlete may actually feel like they're weaponized and they're using their bodies to inflict pain on their opponents. There's a tendency towards a cultural minimization of the injury. Fortunately, this is improving with greater cultural awareness of the serious nature of concussion injury. And there's a lot of conflicts of interest in the assessment of recovery, so that the player wants to get back in the game. The coaches want the player to get back in the game. The teammates want the player to get back to the game. And in the youth athletes, the parents want the athlete to get back in the game. And yet there's nobody that's advocating for the brain health of the athlete. And these conflicts of interest lead to a muddy water with regards to safely managing the concussion and returning an athlete safely to gameplay. And this also is influenced by a certain level of urgency to return to gameplay as well. For our interests in objective physiologic measures that can be applied to concussion management, the one unique feature about sport-related concussion that is most relevant is that it occurs in a controlled environment where the time and place of injury are predictable. The injuries are likely to happen during a game or during practice. And in that regard, one can anticipate ahead of time precautionary measures, which, for instance, like baseline assessments of the athlete with which someone can compare the function of the brain prior to injury to after injury assessments. Now, in our lab, we have previously reported in a group of 60 high school aged athletes that S-Loretta, a QEEG analysis, demonstrates a broad spectrum deregulation in high school age athletes soon after concussion. This was within about a week or two after their concussion. This deregulation was predominantly an increase in current source density amplitude, and there were significant gender differences in this deregulation. Females demonstrating significantly increased beta gamma activity in comparison to males. Males demonstrating significantly greater beta deficiencies than females. And this deregulation was seen persisting beyond clinical resolution of the concussion injury. There were significant correlations between S-Loretta deregulation and performance on a computerized neurocognitive test battery, which was region-specific and task-specific in a dysfunctional relationship. In other words, the more the deregulation, the worse the performance on the cognitive test battery. With the exception, and that was in all frequency bands with the exception of the alpha band, in which a compensatory relationship, in other words, increasing deregulation in the alpha band led to better performance on the concussion cognitive test, and that was seen exclusively in the alpha band. So currently we propose an integrated model of assessment that computerized neurocognitive tests are currently widely used to establish baseline cognitive performance in athletes at risk for concussion injury. And this is these tests are applied prior to the start of the season so that an adequate baseline is established. And then they're subsequently used to compare in post-injury performance after the injury has occurred, and statistical comparisons can be made between post-injury and pre-injury baseline. And this can be used to assess readiness to return into gameplay. Now, these assessments, however, can be skewed by subjective effort. For instance, some athletes will feign poor performance at baseline in order to be able to perform at baseline after injury. Or some athletes may not really be motivated to return to the game and may feign poor performance on the after injury assessment. So there can be some subjective effort issues with regards to performance on these tests. And this is why an objective measure of brain performance is desirable in this setting. I want to give the background of the Excellent Brain Computerized Neurocognitive Performance Test. It is a test battery which consists of several subtests. The first subtest is the word memorization test, in which a series of 15 words is presented one at a time. And then the subject has a forced choice of one word or another as being on the list that was presented. And the right arrow key or the left arrow key are used. This is repeated 3 times at the beginning of the test. Second part is the emotional identification test, in which pictures of people expressing emotions are presented one at a time, and the subject is to press the space bar every time they see a new face, unless it's a target face like happy, sad, angry, afraid, or neutral faces. So the test is repeated for each emotional target. Tapping test is a simple psychomotor tapping test in which the spacebar is tapped as fast as the individual can within a span of 30 seconds. This is followed by the time estimation task, in which a target time is presented to the subject, and the athlete or subject is supposed to tap the spacebar once to start their clock internally, and then tap the spacebar a second time when they feel the target time period has elapsed. The stop or go task is presented in 2 different versions. In the first version, the word stop or go are presented one at a time, and the subject is asked to tap the spacebar when the word stop appears, and not when the word go appears, and not when the word stop appears. In the second iteration, the subject is to tap the spacebar when the color of the word is blue and not when it's red. The one-back test is a test of memory of photos as they're presented one at a time. The subject taps the spacebar every time they see a new photo, unless it's the same as the photo prior to that presentation. The sentence completion task is a task in which sentences are presented one word at a time. And the last word in these 5-word sentences either makes sense or doesn't make sense. The right arrow key is tapped when it makes sense. The left arrow key is tapped when it doesn't make sense. Towards the end of the task battery, the facial recognition task is pairs of faces are presented, and the subject is to indicate with the right and left arrow key which of the faces were presented earlier in the task battery. And finally, there's a 4th iteration of the word recognition task. Pairs of words are presented. One of the words was on the list that was to be memorized at the beginning of the test battery. The word list is not presented another time, and the subject indicates with the right or left arrow key whether the whether the word on the right or the word on the left was on the list. Now, as you've seen in the construct of these computerized neurocognitive tests, that they are well suited to provide stimuli for event-related potentials. Computerized neurocognitive tests are typically controlled or are constructed with controlled stimulus presentation and require a button click or a mouse click for responses. These responses are subsequently analyzed for accuracy and reaction time. But it's very simple to include in the source code of these computerized tests code that marks a simultaneous EEG recording, which is easily embedded in the source code, and then that can subsequently allow for analysis of the EEG for event-related potential processing. And this provides an added layer of objective measurements in the baseline and post-injury testing in sport concussion management. These are the tasks as we have just looked at them. And these are the potential evoked potentials that can be evoked by these. For instance, the time estimation task potentially can evoke a Bright Shaft's potential. In the go, no, go tasks, you can elicit P1, N1, P2 complex in the occipital and posterior temporal regions and a P300 for task execution. Same with the facial emotional recognition. The facial recognition can elicit a P1, N1, P2 evoke potential, as can the word immediate and delayed memory task. The sentence completion task can invoke a mismatch negativity or N400, and the one back immediate visual memory test can elicit a P1, N1, P2 complex. Of note, the facial emotional expression can also elicit a P170, which is typically maximal in the right frontal and temporal regions. So here we have an example of how this can actually be applied and executed. This was a subject who was doing the facial emotional recognition task, and simultaneously the source code was sending a mark out to the EEG. So with each stimulus presentation, so that the EEG could subsequently be analyzed. This first image is the evoked potential measured at PZ with a spectrogram of the individual trials over time with potential. So each line, each of these voxel lines is one trial or one stimulus presentation for the target emotions. And the targets are divided in presentation. So the first 8 or so targets are for happy faces. The next are for sad faces. The next group is for afraid faces. The next group is for angry faces, and the final group is for neutral faces. So the stimuli stay the same, but the task requirement for vigilance for a particular emotion changes. And this display shows all of the presentation of targets for all emotions. Notice that at PZ, we really don't have much in the way of an appreciable evoked potential. There may be some generalized electronegativity in the later portions of this evoked potential measured at PZ. But for the same task measured at FP2, the right frontal lobe, we have a nice, brisk, positive electrical potential occurring at approximately 170, 180 milliseconds out, followed by, again, an electronegativity. The interesting thing that I find about this particular subject trial, it's an N of 1, is that there are distinct differences between the ERPs that are elicited by the different emotional tasks. So happy faces will elicit a different emotion than the target of the SAT. Remember that the stimuli are the same, but it's just what the individual is vigilant for that will change the ERP patterns. So in summary, the concussion is a common brain injury, particularly in sports. And in sport, where the environment is controlled, concussion occurrence is predictable in time and location. Currently, measures of brain physiology are not indicated in guidelines for sport concussion management. And preseason evaluations to establish baseline brain performance, including computerized neurocognitive testing, has become a standard of care. Computerized neurocognitive tests can easily be adapted to serve as stimulus generators for ERP recording and analysis, adding a low-cost and time-efficient layer of objective measures of brain performance to aid in the management of sport-related concussion. Thank you.